This program contains graphic images and discussion of medical procedures. Viewer discretion is advised.
Thank you for the invitation. I'm trying to summarize in this less than 1 hour or so some of the work we have been done in the last 2 decades, probably, with the use of epithelial stem cells. As you can see here, you see these colonies. The colonies are made by clonogenic keratinocytes. No matter whether you isolate them from skin, from the cornea, from the conjunctiva, those are clonogenic keratinocytes, each colony being the progeny of a single clonogenic cell. Eventually, this colony will grow and will fuse. And once they do that, they generate in vitro a cohesive sheet of an epithelium. In this case, you see is an epidermis that's been grown on plastic. More recently, also thanks to the original work of Yammer and Don a few years ago, we grow these cells for clinical purpose, just for clinical purpose, on fibril support that makes the life of surgeons actually much, much easier. Now, these cultures originally developed in the 80s by Howard Green in Harvard. In those days, I was in his lab. Has been used for many years for clinical application in skin burns. And this is already history. Today, I'm going to focus on 2 clinical applications that we developed in the last years. One is a cell therapy application for the ocular burns. We'll dedicate a little bit of time, but most of the time is dedicated to the gene therapy of this disease called epidermolysis bullosa. So let me just summarize the work that we have done on the ocular burns that has been developed in these years by specifically and mostly by Graziella Pellegrini. We have been working together since over 30 years, and in collaboration initially with Panorama, who is a corneal pathologist at the San Raffaele Hospital in Milan. And make a very long story, very short. I mean, our ocular surface is covered by the cornea and the conjunctiva. This area is called the limbus. Now, in the basal layer of this rim of epithelium, called the limbus, are located the stem cells of the corneal epithelium, that are these cells here. We have devoted a lot of time in work in characterizing these human limbal stem cells. We showed that they've been located in the limbus. We've shown that their proliferative regenerative potential is regulated specifically by a transcription factor called delta N PCC3 alpha, together with another transcription factor, CBP delta, that can regulate their self-renewal. Now, these stem cells, they generate their progenies, blue cells, that are progenitors that made up the corneal epithelium. If we have a burn in the ocular surface that affect the cornea, but not the limbus, then the limbus gets activated and is able to regenerate the corneal surface, the corneal epithelium. However, if we have a severe burn in the eye, where we have destruction of both the cornea and the limbus, the conjunctival cells will take over, will cover the ocular surface, will take then vessels like this, because you remember that the cornea is not vascularized directly, and what we have, what is called a corneal opacification. In this case, where we have corneal and limbal destruction, the corneal transplantation that usually is a surgical procedure that can be used for lesions where the limbus is preserved cannot be applied because we have not enough stem cells to cover the graft. So it is very difficult clinical situation. Now, this is the eyes I'm talking about. You see, this is complete opacification. There is basically a limbal stem cell deficiency. There is a pannus. These patients, they have visual loss, obviously, but they have also severe symptoms in corneal opacification. During the years, what we have developed for unilateral lesion, but also for bilateral lesion, is the possibility, starting from a very, very small limbal biopsy, about 1 millimeter, to identify those stem cells. And we'll go back to the different clonal types at the end of my talk, to regenerate the corneal epithelium, to transplant the corneal epithelium, which has been regenerated in vitro, onto this area. How? I'm going to show you the surgical procedure, which is not a very pleasant movie. So for those of you that don't like surgery or they are quite sensitive to surgical images, be aware that it's not a pleasant movie. This is the biopsy, about 1 millimeter biopsy that is taken from the LTI, and this is the biopsy from where we grow these cells. What the surgeon now, what Panorama is doing now, is to remove, this pannus, to remove this conjunctivalized epithelium, and to expose the corneal stroma. Onto this corneal stroma, then we can transplant the limbal cultures that eventually will engraft and will regenerate the corneal surface. The clinical results that you can get with this technology is the following. You see, those are eyes that I was referring to, completely opacified, vascularized, with severe symptom loss of vision. In this case, for instance, you can see here there is the sign of a keratoplasty that failed because there was not enough stem cells there. But once you regenerate the entire physiology of the cornea, that's what you get. You have a complete, total restoration of the cornea with no vascularization anymore. The cornea gets transparent. In the very long term here, those are eyes at 10 years follow-up, but now we have over 20 years follow-up, and you have a full recovery of visual acuity. And you can do that also when you have bilateral lesions. About 15% of these patients, they have bilateral lesions. That is a much more severe situation because these patients are basically blind. This, for instance, is the case of a young man that received alkaline burn in both eyes, and he gets lots of corneal transplantation that failed because of the lack of the limbal stem cells. But this area of the limb was quite good. So from one small biopsy, we were able to grow both grafts, and this is the full recovery of both eyes in these patients with full recovery of visual acuity. These patients was not able to drive or to work, and now is running a normal life. This is basically all I wanted to say about this because it's all published. Just want to mention that after many years of application, we were able to get a formal approval by the European Medicine Agency, by EMA, so at least in Europe, of this advanced therapy with the name of ocular since February 2015. And right now we are collaborating with many countries in Europe for the application of this therapy. Actually, the main reason why I wanted to show this is that what I'm going to show from now on, so all the gene therapy is not coming out of the blue, but is coming from many years of work, both on the basic biology of epithelial stem cells and on what we have learned in terms of cell therapy in terms of tissue regeneration. So it's only after all this is completed that we can really think of modifying genetically those cells to make to have a gene therapy approach. And in our field, there is a really devastating disease, which is called epidermolysis bullosa, or the butterfly children disease. For those that are not aware of this disease, which is very rare, is genetic disease, our epidermis is attached to the underlying dermis through these black structures, which are called emidesmosomes, with the basal lamina. Those are the emidesmosomes. It is a chain of protein starting from the keratins in the basal keratinocytes, mainly keratin 5 and keratin 14, they have interaction with the specific integrin at the emidesmosome, which is called integrin alpha 6 beta 4, that gets in touch with the laminin 5, also known as laminin 332, that in turn gets it is connected to the collagen 7 anchoring fibrils in the dermis. This is how our epidermis is kept in place. Now we can have mutation in each of the gene coding for these proteins. If we have mutation in keratin 5 and keratin 14 in the simplex form, the blistering, the disruption of the junction occurs within the epidermis. So within the epidermis, and usually those simplex forms are dominant, but they are, in general, they are less severe than junctional and dystrophic, and they tend to get better during time, although there are also severe forms of simplex EB. The most severe forms are the junctional and the dystrophic. In the junctional form, mutations are usually in one of the three chains of the heterotrimer laminin 5 or the alpha 6 beta 4 integrin or collagen 17. And as you can see here, the blister is within the lamina lucida of the basal membrane. So here we have a detachment of the entire epidermis and we have bad blisters. If the dystrophic type, the lesion is more deep, so it's below the basal lamina and the superficial dermis, and because it is due to the mutations in the collagen 7. The disease is devastating because these patients, they are born with blisters, usually in the generalized form. They are born with blisters all over the place, all over the body, spontaneous blistering or upon minimal trauma. So their quality of life is very, very bad. Think about that about 40% of patients with the generalized junctional EB, they die before the age of 18. Some of the patients, they have a double knockout mutation, for instance, in the beta 3 chain of laminin 5, they die within the first few months of life. Those patients, they get to adulthood, so they mutations that allow them to survive, they have a very, very poor quality of life, and also a short life expectancy because they have squamous cell carcinoma. There are many palliative therapies that can be used, or medications to try to somehow control these lesions. But the real breakthrough could be a gene therapy, so the genetic correction of the disease. Many years ago, we did the first phase 1 phase 2 clinical trial ever made on the junctional EB, on the patients that have mutations in the LAMB3 gene of the heterotrimer using retroviral vectors, and those genetically corrected epidermal sheet were transplanted on the upper legs of this patient. And as you can see, at now our 16 years follow-up, the epidermis, the skin of this patient has been completely restored. And as you can see, here the epidermis is transgenic, as you can see from this in situ immunization. And the expression of laminin 5 was very low, if any, at admission. And after the transplantation and transgenic skin, you see there is normal expression of laminin 5. So there was a first proof of principle that the ex vivo gene therapy could be done. And at that time, we were ready to treat more area of this patient and more patients. But we had to stop all our activities, the cornea and the gene therapy, because meanwhile, in Europe, all the regulation for advanced therapy changed completely. We had the new regulation, the regulation number 1397 of the European Community, that was considering this something that could not be done as we done before at an academic level, but they were drugs. So we have to have a GMP facility, we have to have a GMP certified facility, we have to go through formal phase 2, 1, 2, 3 clinical trials. And at that time, we did not have any of these things. So we have to stop, to build up a new building, to have a GMP certification. We have dedicated much time at the beginning. We have to create a biotech company, all of them, because our, the university is not able to follow these rules that are made for pharmaceutical companies. And initially, all of them was dedicated to the cornea, because the cornea was the therapy that was ready to go onto the market. So we were able to start our gene therapy only in 2015, after 9 years. And what we did in collaboration with John Bauer in Salzburg University was to reproduce what we have done with Claudio, with this first patient, this time under GMP condition. And as you can see here, this is a large area with junctionally B due to LAMB3 mutations, exactly as Claudio. And there was not healing since 10 years, and it was completely restored by the transplantation of the transgenic skin. So at this time, we were able to start our trial again. But something happened to us that changed completely our strategy. Phase 1, then stop, make phase 2, then stop. What happened to us was the story of that you probably know of this kid, Hassan, that was a 7 years old boy, at the time was a 7 years old boy with a very severe form of junctional EB. Due to an homozygous mutation in intron 14 of the LAMB3 gene. This kid was living in Syria. So under the civil war, a patient with junctional EB cannot be treated, very difficult situation for a patient with a very fragile skin. So the family moved to Lebanon, but the situation of the kid was getting worse. So through humanitarian organization, they moved to Germany as refugees. The kid was admitted to a dermatology department in Germany, but meanwhile he had a super infection of Pseudomonas aeruginosa Staphylococcus aureus. So instead of having the blisters that come and go in the skin, Hassan was losing the skin completely. So they have to move the skin to a burn unit because the amount of skin loss was too big. And he was admitted to the burn center in Bochum in Germany. And I have to tell that all the clinicians there, they try their best to save the life of the skin using all the tools they have for burn patients. But Hassan was not a burn kid, it was a genetic disease. So all the therapy failed. So meanwhile, he was in deep sedation, so in induced coma for few months, about 4 or 5 months. He was in critical condition. The prognosis was very poor. So they called us to see whether we could include Hassan into the clinical trial. It was not possible because he did not have any of the inclusion criteria to be included in a phase 1 or phase 2 clinical trial. So what we decided to do was to ask the German regulatory authorities for a compassionate use. And I have to say that the regulatory authorities in Germany, they gave us an answer in 3 weeks, so very fast. They said, okay, try and go ahead. So what we did, was to take a 4 centimeter square biopsy and to generate about 1 square meter of transgenic epidermis. It was a very difficult task because it was the first time that we make such an amount of epidermis, try to control, after gene correction, try to controlling the right balance, and I will be back to this, between the stem and the progenitors. And we did this, we transplanted this transgenic skin in 2 big operations. One operation was done for the 4 limbs, and the second operation 1 month later for the entire back. This is the preparation of the wound bed where you expose the dermis, and then is the application of the grafts. And let me show you what happened. This is the left arm, you see completely covered by the grafts, and by 10 days, 2 weeks, at the removal of the basilicos, at the removal of the bandages, you see how nice is the new epidermis that engraft and took. And for me, it was a very, I have to tell you, very strong emotion to see how nice was the epidermis in this difficult situation. And by 1 month, the entire arm was covered by new form epidermis. Same thing was for the left leg. You see the left leg was transplanted. And by 10 days, 2 weeks, 1 month was completely covered of skin. The same thing was for the right leg. Here we had a problem of bleeding. So as you can see, at 1 month, we have total regeneration of the epidermis, but in this area where we have bleeding, so we have to go back with some more grafts in these areas. But by transplanting the 4 limbs, the situation of the kid was much better. So he was reacting by covering his skin. So 1 month later, we were still in coma, actually, in induced coma. That was, by 1 month later, we transplanted the back. Look how bad was the situation of the back. But the work that Tobias Hirsch, the surgeon, did for the preparation of the wound bed was excellent, because as you can see, here you have this quite nice receiving bed for the grafts with this dermis. This is the application of the grafts. And by 1 month, you see the entire back of Hassan was covered by skin. We did not have a good take in the buttocks, and is a problem that we historically had also with the bird. For some reason, the engraftment in the buttocks is not very good. But you see there are islands of keratinocytes here. So what we did was we just let the cells do their job and keep changing the medication. By 4 months, the entire back was covered by epidermis. So the bottom line of the story, that Hassan was admitted with a very poor prognosis in the hospital, and in June 2015, and before Christmas, he was out of the coma. He was discharged from the hospital in February 2016, and there was the visit he did in Modena in 2017. There was a present of a small Ferrari that he gave to us. You know, Modena is the town of Ferrari. And 1 year later, so in March 2017, you see the regeneration of the epidermis. Every single biopsy we took at 4 months, 8 months, 2 years, you see that the in situ hybridization is positive. So it's entirely transgenic, the epidermis. This is the absence of laminate 5, the meta-3 chain at admission. This is the way it's supposed to be, the normal control. And you see the expression of normal amount of laminate 5 in every single biopsy we took. And by transmission electron-microscopy, you can see that we have a normal number structure of epidermosomes. So this is why Hassan now is running a normal life. This is his appearance at 3-year follow-up. This is his appearance at 5 years follow-up. The area that we have not transplanted, because we transplanted about 80% of his body, but about 20% was not transplanted. They're still making blisters, but all the transplanted area has been regenerated. And even after 5 years, that for the skin is a very long-term follow-up. We renew our epidermis every couple of months. So 5 years follow-up is a very long time because of the renewal. Again, the more the time, the better the epidermis looks. But most importantly, you have transgenic epidermis and expression of normal level of lamellar 5, which is comparable to a normal control. We have analyzed in the last, the paper just came out, actually, on the 5 years follow-up, every single parameters of the epidermis. We have normal appearance of the epidermis. We have normal expression of the involucrin, for instance, or keratin 10, that are markers of differentiation. We have normal expression of keratin 14 in the basal layer. We have presence of Langerhans cells. We have melanocytes. We have Merkel cells. We have P63, that go back to this, is quite important. P63, as I said, for the cornea, also for the skin, is important for the stem cells maintenance. The only real difference we saw as compared to a normal skin is that there is a decreased amount of elastin and of elastin fibers. That probably has nothing to do with the grafts itself, with the transgenic epidermis. It's to do with the years of inflammation or fibrosis, sometimes inflammation, that the patients had during his initial 7 years of life. And also the wound healing is normal, as you can see. So the bottom line of the story is that we did not see a single blister develop in the transplanted area. We have normal wound healing upon injury. We have no adverse event. Despite the amount of skin we transplanted, we have no pain, no itch. We have normal mechanosection, normal nociception. We have no need for ointment or creams. We have dyschromia, as you saw from especially in the back, and we can go back to this if you want to do later in the discussion. There are reasons for the dyschromia and a decreased amount of elastin fiber. Somehow, Hazan was paying us back because he was very instrumental, I should say, in trying to understand more about the biology of our epidermal stem cells. In which sense? Well, there was some debate still in the literature whether the mammalian, this experiment was mainly done in mice, obviously, but by lineage tracing and all sorts of technique, whether the mammalian skin is sustained by a population of stem cells and by transient progenitors and by terminally differentiated cells, as in the hematopoietic system, or if we have a single class of a progenitor that stochastically give rise to suprabasal terminally differentiated keratinocytes, or whether we have a population of resting and activated stem cells, you can also call committed progenitors, that give rise to the terminally differentiated cells, or if we have a mixture of this, where you have a long-lived stem cell, a short-lived or activated stem cells, then you have progenitors, and then you have terminally differentiated cells. Well, since the epidermis of Hazan is transgenic, we have the tool now to try to solve this issue, at least with the mammalian, with the human epidermis. And in order to explain this, I have to go back to the different type of clonogenic cells we have in the. If you take those clonogenic keratinocytes I showed you at the beginning, you can isolate those clones, and you can identify 3 types of clones that are called holoclones, meroclones, and paraclones, originally identified by Baradoni Green in 1887. For many data, you can distinguish those clones by the level of expression, not the expression itself, but the level of expression of delta and Epithelial alpha that is very highly expressed in holoclones and progressively decreased during clonal conversion. We have evidence that the holoclones, we have the stem cells of the squamous epithelia because of indirect evidence, not direct, indirect evidence, showing that, for instance, if you lack those holoclone-forming cells, you don't have long-lasting epidermis. But again, that was a sort of indirect evidence. Now, we have the tool to see whether this is true or not. How? By clonal tracing. As I told you, every single clonogenic keratinocytes give rise to a cornea. When we make gene therapy with retroviral vectors, we have a random integration of the transgene into the genome. And the chances that you have the same, very same integration in two different clonogenic cells is almost impossible. So by studying the integration, by analyzing the cells at the single cell level, we can analyze different clones with identified by the specific integration. And those are the clones that once they are together, they make up the epidermal sheet that you transplant into the patient. And as I said, you can analyze this at a single cell level by make clonal analysis. So you can take every single clonogenic cells, put them in a dish as a single cell, isolate the clones, and by making the clonal analysis, identify those clones as holoclones, meroclones, and paraclones. But this time, the holoclone, the meroclone, and the paraclones, they can identify because of the cloning of the integration. So those are the graphs that we put on the patient. And what we show, what we discovered during time, those skin, it gets to monoclonality towards integration belonging to the holoclones. And how did we show this? Because if we make a biopsy of the regenerated skin, let's say 4 months or 8 months, and we analyze the regenerated skin at the single cell level by making clonal analysis, what we found is that we do have the same population of clones, holoclones, meroclones, and paraclones, but all of them, they have the same integration of the founding holoclones. In other words, during time, and we can see this through the integration of the meroclone and the paraclone, they go down, they are lost, but not the integration of the holoclones. They stay stable during time, and during the epidermal regeneration, they give rise to new waves of meroclone and paraclones that originate from the holoclones. So this clonal tracing gave us an unambiguous proof that the human epidermis is sustained by a population of resting and activated, can be activated, long-lived stem cells that they generate short-lived progenitors that eventually generate thermally differentiated keratinocytes. But now we can formally prove by direct evidence that those stem cells are the holoclone-forming cells because they are the only cell type that we find during the regeneration and the time regeneration, and that the meroclone and the paraclones that we can isolate also from directly from a skin biopsy are basically short-lived progenitors. So I told you at the beginning that we was changing our strategy because with this tremendous amount of data in our hands, we went back to the EMA and asked a protocol advice because the disease is very rare and is very severe. So by putting all the data together, the EMA was agreeing in sort of jumping the phase 1 and phase 2 trial and go directly to a phase 2, phase 3 trial, try to speed up a little bit the approval of this therapy. And in these weeks, in these months, at the beginning of 2022, we will start a multi-center European phase 2, phase 3 clinical trial in collaboration with a center in France, Germany, and in Italy. Meanwhile, we will try to tackle also the recessive dystrophic EB using the very same approach. But it's a different disease because it's a different gene, it's a different blister, it's a different situation. And we did this in collaboration with John Bauer in Salzburg. And we have a phase 1 and phase 2 trial, which is still unpublished. And the bottom line of this initial outcome is the following. So it's not as good as junctional EB. Instead of having a 1-year follow-up, a complete restoration of collagen 7 expression with a mosaic, which is no good. And if we make a laser capture PCR analysis on the positive section, we do see the transgene. But if we do laser capture PCR analysis underneath the negative area, we have no transgene. And the same thing is with the anchoring fibers. So the good news is that we have no adverse events, and we do have long-term collagen 7 expression. The bad news is that we have mosaic expression and mosaic epidermal restoration. That for a complicated therapy like gene therapy is not satisfying, at least to me. We want to have the same result as we have in junction. It is true that the patient is telling us that they have less blister, they heal faster. So overall, the situation in transplanted skin is better, but didn't change substantially the nature of the lesions in the skin. So we have to understand why this happens, why it doesn't happen with junctional EB. In order to do this, you have to go back to the lab, to basic science. And I try to summarize in the next few slides 2 complicated and long stories that probably gave us the answer. What we discovered is that in order to understand this, we have to go back to the junctional EB. And this was the work done by Laura De Rosa and Alessia Secone. What they discovered is that the interaction of laminin 5 with integrin alpha 6 beta 4 is instrumental in keeping YAP in the nucleus. And nuclear YAP is essential for the maintenance of epidermal stem cells. In fact, during clonal conversion, we have a decrease of total YAP, you see, almost superimposable to p63 alpha. But if you go a little bit more in detail, you see the nuclear YAP, which is the active YAP, is a much better, identify oligoclone much better than p63, is basically negative metaclone and paraclone, while cytoplasmic YAP, which is inactive, increase during clonal conversion from oligoclone to paraclone. And in fact, if we downregulate YAP, we do not abolish the progenitors, but we abolish only the oligoclones, only the stem cells. And if we upregulate YAP, we block the clonal conversion and we keep the oligoclones stable. So it's essential for epidermal stem cells. And the YAP pathway is altered in the junctional EB because of this interaction, laminin 5, ROC6 beta 4. In fact, if you look here, this is recessive dystrophic and junctional EB. In junctional EB, we have downregulation of YAP, of TATS, and of p63. And by gene therapy, we can rescue this. You see, this is junctional EB at passage 2. We have already a decrease in the percentage of oligoclones. These red bars are the oligoclones. By passage number 6, the stem cells are completely gone. But if we do gene therapy, then we keep and preserve the stem cells. So in junctional EB, we have cytoplasmic localization of YAP, progressive stem cell depletion, and if we do gene therapy fast enough, we can rescue epidermal stem cells. And you can see this also in vivo. This is Claudio and this is Hassan. You see both in Claudio and in Hassan, we have YAP was basically cytoplasmic, but after gene therapy, we have nuclear localization of YAP and restoration of also of P63. And we went on in trying to understand what was the mechanism underlying this interaction by doing single-cell RNA-seq analysis. This was made again by Alessia and by Elena Enz in collaboration with all the bioinformatic people in my university by doing single-cell analysis. And I don't want to come to bother you with all the different clouds and population of cells that we have identified, holoclone, monoclone, paraclones, and the clonogenic markers, the holoclone markers. We did a lot of work on this. What was important, what we found, that downstream of YAP, there's a transcription factor, FOXM1, that again is expressed in holoclones and not in monoclone and in paraclone, that reproduce what is happening with YAP. By FOX ablation, we have ablation of holoclones, but not of the progenitors. And by enforced FOXM1, we don't create new stem cells, we don't create new holoclones, but we stop clonal conversion and we preserve the population of stem cells of holoclone-forming cells. And again, in junctional B, this is what happens. In junctional B, we have decrease of the holoclones by both gene therapy or overexpression of FOXM1, keep the stem cells at a normal level after gene correction. So we have YAP, FOXM1 dependent stem cell depletion in JED that is rescued by gene therapy. This pathway here is working in junctional B, but it's not working in dystrophic B. So what we think is going on in the mosaic problem that we have in dystrophic is a competition problem. In other words, when we do gene therapy with the LAMB3 gene, since we rescue the long-term repopulating stem cells, at the end of the story, the entire epidermis will be formed by the genetically corrected cells. But this does not happen with collagen 7, where we do still have a competition between transduced and untransduced cells. So in order to solve this, we had to go and do again at the clonal level and try to understand what really we were transplanting on these patients in the first initial, I should say, a little bit probably too optimistic phase 1 and phase 2 trial. And with the vector we were using, we had approximately 75% efficiency in collagen 7 gene correction, which is quite high, but it's not 100%, which is difficult also to have 100% because collagen 7 is 9.2 kilobases. On the top of this, collagen 7 CDNA is quite a difficult gene to manage because it has a lot of collagenous repeats. So it is easy to have recombinants. So we analyze those recombinants at the clonal level, and what we discovered by analyzing all the single clonogenic cells, because obviously the potential recombinants, you cannot see this in a mass, in a bulk population. You have to go to clonal analysis. We saw that approximately 25% of the clones that were forming the graft contain recombinant collagen 7, not full length, and only about 50% of the clones were containing full length collagen 7. So we have, let's say, about 50% gene correction. So we decided to stop the trial, taking out of the trial the good that we could take. So no adverse event and the possibility of having a long-term expression of collagen 7. Change completely the vector. Designed a new SYNGAM retroviral vector that is now over 95% efficiency, but we change also completely the way of transducing the cells by doing 2 rounds of division, by the combination of a higher efficiency of the C-gamma retroviral vector together with a 2 rounds of infection. Now, every single clone that we analyze at least get 1 full-length copy of the collagen 7. The price we are paying from this is the number of vector copy number, because in the old vector we have approximately 1.5 as an average vector copy number per cell. We have approximately 4. But again, this counterbalanced by the fact that in principle, this is a C-gamma retroviral vector and not a classical old-style retroviral vector. So with this in mind, we think that at least the problem of the competition should be solved. And so we expect that by doing the trial now with collagen 7, using this newly developed vector, we should be able to eliminate the competition and to have a regeneration of the epidermis with complete restoration of collagen 7 as it happens with the laminate 5. And with this in mind, this year, 2022, we will ask for a new clinical trial, again, multi-center European clinical trial with Italy, Germany, Austria, and France, using this vector to avoid cell competition. It's basically all I wanted to tell you, hoping that you got the main message, especially for the young people, for the PhD students, the message that you need to combine a lot of basic science if you want to do proper regenerative medicine using appropriate stem cells with appropriate definition and characterization of the type of cells that you are using. And this basically was happening with the hematopoietic system. The spectacular results that people are having with the gene therapy of hematopoietic cells is because there is a lots of basic background in the hematopoietic system. I just want to finish by telling you that we want to approach the problem of the butterfly children 360 degrees. Everything I showed to you today, it can be applied to recessive forms of the disease, compound, heterozygous compound, recessively inherited form of the disease, not for the dominant. And about 50% of EB, they are dominant, especially the patients with simplex EB. But within the family of simplex EB, there are patients that are severe forms of EB. So you cannot do gene addition these patients because the mutation is dominant. So in this case, we have already evidence, still unpublished evidence, that we can use gene correction, so gene editing using the newly, everybody knows, CRISPR-Cas9, to make up a transgenic epidermis where we have gene correction instead of gene replacement. And I want to finish this by mentioning the people that actually this slide is more dedicated specifically to the story of Hassan, that, believe me guys, was a really enterprise, because for months all the people in the Center for Regenerative Medicine were completely devoted to try to solve the problem of Hassan. Special thanks go to Graziella Pellegrini, that she was extremely useful for the translational part, especially in the operating room together with the surgeons. Sergio Bondanza is a technician that works with me since almost 35 years now, and he was the guy that was growing the cells of Hasan, Laura, Elena, Roberta, and Alessia would analyze all the cells before and after grafting. And the collaboration with the group of Silvio Micciato, Oriana, and Mattia that was together with Michele Morgante and Davide Scaglione, and also Enrico and Elena, were helping us a lot in the clonal tracing of the study of the integrations, the clonal tracing, the identification of different clonal types based on the site-specific integration of the transgene. And obviously, the clinical group of people in Bochum, Tobias Hirsch is the surgeon that did all the together with us on the transplantation. Although I want to draw your attention to Tobias Reuters, because this is what Tobias Reuters did is amazing actually. If you go to the supplementary appendix of the paper and you read the clinical history of those months, where it is amazing how Tobias was able to balance everything to keep him alive in very dramatic situation. So both Tobias Hirsch and Tobias Reuters was very, very important in doing this with the assistance of John Bauer for the dermatological point of view. And with this, I thank you for your attention. I apologize for my Italian accent. I hope you understood everything. And I'm ready to take any question.
That was spectacular and very inspiring. And I think you made an excellent point that that even if what you want to do at the end of the day is to help patients, it still requires a foundation in fundamental basic science, fundamental developmental biology, and stem cell biology. And you've made that point admirably. That's great, and a great take-home lesson for all the biologists who attend these seminars. So anybody who has questions, please put it in the Q&A. We have a time for a few questions, and I'll pitch them to Michele. The first question that's come in is, obviously, to be able to do your transplants, you require a lot of skin and monolayer, so to speak. How long does it take to grow those sheets for the transplantation?
Well, those cells are really spectacular. And we have a lot of experience with the birds. It's quite fixed time. It is approximately 3 weeks. So from the moment you take the biopsy, from the moment you have a graft ready to go, at this point, it doesn't matter whether it's 1 graft or 10 grafts or 50 grafts, because it's just the second step, amplification step, that is different. In any case, it takes about 3 weeks. In the case of gene therapy, you need sort of a week more, because you have to introduce the step of gene correction. So, for instance, for the cornea is about 3 weeks. For the gene therapy is about 4 weeks from the moment you take up the biopsy.
Great. All right. Another question is, and you've answered this partially, but you talked a good deal about how various clones can outcompete each other, and how dystrophic EB was a little bit different than Claudio with junctional EB. How is it that, the question is, how is it that your donor epithelial stem cells were not outcompeted by Claudio's own epidermal stem cells? Or did they outcompete those? Why did not his own endogenous flawed outcompete?
Your question is why the transplanted area in the leg of Claudio did not expand?
No, why? I think the question is, obviously he's generating his own epithelial stem cells that are genetically flawed, did your transplant suppress them, outcompete them? Since
Well,
you didn't...
no. You see, when you transplant this, you remove completely everything. It's like in the hematopoietic system.
Okay.
If you want to do a bone marrow transplantation to transplant the hematopoietic system, hematopoietic stem cells for the leukemia, for instance, you have to do myeloablation. So you have to make room.
Right.
This is what we do. So we remove everything. So what you transplant is your graft. So in this moment, there is no competition.
Okay.
And in the junctional limb, there is no competition for two reasons. One is that because the efficiency of transduction, it is very, very high. It's over 90%. Second, because in any case, the transduced cells, they have a selective advantage on the wild type cells.
Okay.
With the dystrophic, you are putting a graft where already in the graft there is a competition. You see, that's the problem. What is strange though, and we didn't explain this, is the opposite. So we were expecting that the transplanted area in junctional limb, they would expand laterally and substitute the region of the junctional skin flanking the transplanted area. But does not happen. Even after 15 years, we still have precisely the stamp, the real area that has been transplanted, and the cells are not moving out, probably because they sense that there are keratinocytes there and they don't invade the remaining part of the skin. We were hoping actually that it would happen in junctional limb, but it doesn't.
Okay. The next question is, with overexpression of the FOXM1 gene, does it reduce the differentiation of the transgenic stem cells? I guess, does it alter the differentiation, the transgenic stem cells? Is their differentiation fate altered in any way?
No, really. I mean, what we found by sustaining the expression of FOXM1 is that we simply stop clonal conversion. We did not create new stem cells. You see, when you keep growing those in serial cultivation, you have during time a progressive depletion of holoclones, and at the end, you will have a replicative senescence because all the clones become monoclone and paraclones. And at the end, you have an exhaustion of the proliferative potential. It may take 6 months or 20 passages. Usually, before getting into replicative senescence, the cells, they do 150, 180 cell doublings. By overexpressing FOX, we stop the clonal conversion. So, and the percentage of holoclone-forming cells remains stable during the passages, and the cells keep growing. But the epidermis that is made, from what we can see obviously in vitro, does not have any alteration of the markers of differentiation. It behaves normal. It just stops the clonal conversion. And the same things happen with the aperture.
Another question is, obviously you're doing autologous grafting into the patients. What more globally, what are your immune considerations when you do these procedures? Must it always be autologous, or are there any concerns where you would ever need to try to do immunosuppression or anything at all like that?
Well, there are two aspects of this. One is the cells, one is the transgene.
Mm-hmm.
Concerning the cells, the keratinocytes, forget about using allogeneic keratinocytes. The allogeneic keratinocytes, they are not accepted by the organism. They don't even engraft. People have tried that for full thickness burn. They have tried that also for the severe corneal lesion. You have to forget about the use of allogeneic keratinocytes if you want to have permanent engraftment and long-term regeneration. For the transgene, we have obviously during the trials we have evaluated all the immunological parameters, and we never find any sign of autoimmunity related or directed to the transgene. With laminin 5, there was no sign for this. So we will analyze this also, though, for the collagen 7, of course. For the collagen 7, we will carefully analyze during the trial the onset of the potential autoimmunity. As a matter of fact, at the beginning, we will select only patients that they had a minimal expression of collagen 7. We will not include in the beginning a double knockout, for instance, where there is complete absence of the protein. This is something that we will address a little bit later on. So, try to minimize a potential autoimmunity towards the collagen 7.
Great, thank you. I think we're down to our last two questions, and then we'll let you go. Given that EB is a congenital disease that is present even in the newborn period and is so devastating, do you envision being able to go even earlier and earlier in your gene therapy approach?
Well, I thank you for this question, because this is always a matter of discussion with regulatory authorities. The way I see it is that, yes, the answer is absolutely yes. This is a pediatric disease, and the sooner you treat the patients, the better it is, for a number of reasons. One is that the amount of skin that you have to make is lower because the kid is small. Two, the quality of the epidermal stem cells, by definition, is better. Three, you minimize as much as you can the chronic inflammation, and in the case of dystrophic, also the scarring that you can go through if you treat these patients too late. Obviously, it cannot be too early, because you have to make sure that the patients is suitable for that type of treatment. Remember that these patients, they also have internal lesions. They have problems in the esophagus, in the oral mucosa, in the upper airways. And in this case, in some cases of some form of the disease, those internal lesions are very severe. And so those are patients that probably cannot benefit from the skin. So you have to wait a little bit to see how developed the disease in the first few months or 1 year, 2 years of life. And then, though, to me, once you've done that, the sooner you do, the better it is for the patient, and also for the therapy, for the stem cells, and for everything. And obviously, you don't have to do such a dramatic surgery that we did for Hassan. We were forced to do that gigantic operations because Hassan was dying. But if you can control the disease from the beginning, you can progressively substitute the skin in the smaller operation in, in 1 year, in 2 years. If the kids start developing the blister in the arm, instead of doing just the medication, you do the graft and the arm is fine. Then 2 months later, you do the other arm, and so on, in order to have a less aggressive type of surgery. That's the way I see it at the end of the story.
And this is the last question, and I'm sure you had to answer this for all the regulatory hurdles you had to go through. are there any neoplastic considerations, any concern about down the line there being any kind of neoplastic transformation, particularly from the engineered stem cells?
Yeah, that's we always we are make a big monitor of this. These patients, even though they are transplanted, we have a formally approved trial, which is called observational low interventional study. We are going to follow these patients for decades.
Right.
But let me tell you this. Hundreds of patients that we have treated, hundreds of patients that we treated for the burns with cell therapy, we never observed the occurrence of a squamous cell carcinoma or any type of carcinoma due to the graft. And this is over a 30 years follow-up. For the transgenic cells, obviously, it's probably too early, but remember that we have the first patients with 15 years follow-up, the second one, the last one with 5 years follow-up. And although are only 3 patients, I think that we should think in a different way. We have transplanted on these patients something in the range of 10 to the 8 to 10 to the 9 chronogenic keratinocytes.
Mm-hmm.
So, which is approximately, if I remember correctly, 10 to 20 times more the number of cells you transplant in a bone marrow transplant, in the hematopoietic system. It's tremendous amount of chronogenic cells. And those cells, they renew the epidermis constantly. We never saw anything. We never saw, and in the skin, you immediately see it because it's out. So if there is a problem or a clone emerging or any type of problem, you can immediately see it. It can immediately be removed.
Right.
But we didn't see it. And on the top of this, remember another thing, that these patients, the generalized junctional and generalized dystrophy, especially generalized dystrophic, they invariably develop squamous cell carcinoma. Very aggressive, highly metastatic. Usually, those patients, they don't die when they are kids. They get to adulthood. They die because of the squamous cell carcinoma. So probably, although we have no proof of this, probably we are actually decreasing by putting together everything, by putting together the physiology of the epidermis, the adhesion, probably we're actually decreasing the risk of squamous cell carcinoma.
Okay. With your permission, one last question came in, and it's a little speculative the way you just talked about, because your follow-up obviously hasn't been long enough to answer this question, but the questioner wondered, can the transplanted skin repair itself, and will it undergo all of the changes that one sees as a patient ages, including stretch and in other words, will it now repair itself, but also age as the endogenous skin would age?
Well, yeah. Well, the answer is yes, actually, because you see, we know this from the birds. Because the birds, they have already 30 years follow-up, and they behave like, no, in the birds it's a little bit more tricky because you don't have a real dermis. You have a scar. So this epidermis is over a scar. So it is not easy to judge the aging the way you think of. For the transgenic in the EB, yes, we have not enough timing to see this, except in the first patient. And we will define this during time. I would not expect a big difference, though. I would not expect it, I believe that based on the experience we had in the burns, but the time will tell us.
Right. Well, this has been terrific, Michele. And I think it's also wonderful that you began your talk with an acknowledgement of Howard Green's contribution so many years ago to the entire field of regenerative medicine before it was called regenerative medicine.
Well,
So,
you see, Howard was my mentor for years. I still remember that weekly or monthly call that Howard was giving me for 20 years because he wanted to follow all the clinical application. So it was really a mentor for me, Howard. And I really regret, I mean, that he couldn't see the story of Azan because he died in 2015. So right at the time that we were thinking of doing Azan. So I'm really sorry that he couldn't see this. That is, for him, it wouldn't be very important.
well, he was the intellectual inspiration for many of us in our field. So
Absolutely.
thanks so much, Michele. And this was wonderful. Have a good rest of the day, everybody, and see you next month. Bye-bye.
Bye-bye. Bye-bye.